Coevolution is the interaction between two species over generations, each selecting for the expression of traits in the other species which enhance that species' survival and multiplication. The two species continually impose selective pressure on each other, causing an evolutionary change to each species. This relationship has been seen in mutualistic, competitive, and host pathogen relationships. For the purpose of this video, we will be focusing on host-pathogen relationships. Pathogens evade resistance of their hosts by improving upon mechanisms such as the blockage of antigens, escape from apoptosis, and latency of expression. Meanwhile, the host continues to build genetic resistance to protect itself from the pathogen. This concept is often described as a coevolutionary arms race, appearing to be constantly adapting to each other to survive, similar to an arms race between warring countries. However, this does not occur over the lifespan of one organism, but over several generations, through the elimination of inferior organisms and the selection of superior traits. A common example of coevolution of host pathogen is the European rabbit and the myxoma virus, a pathogenic pox virus. Ever since the 19th century and up until the 1950s, Australia had a large uncontrolled feral rabbit population due to the fact that these rabbits had been released by British colonists as a source of meat or hunting, but had no natural predators. The large rabbit population was decimating farmers' production of crops, and thereby hurting the country's economy, as well as forcing many native species into extinction. It was regarded as one of the greatest animal pests the country had ever fought. After several propositions, a drastic decision was made. They would release a virus that would control, perhaps even exterminate, the rabbit population. In late 1950, the standard laboratory strain of the myxoma virus was released into the Australian population. Myxoma contains a large linear double-stranded DNA genome consisting of 159 unique viral genes, with diploid terminal inverted repeats, TIRs. Each TIR contains 12 viral genes, 9 of which encode diverse immunomodulating proteins, giving a total of 171 genes. The viral genome is encapsulated within a brick-shaped virion, and the complete replication cycle of myxoma takes place exclusively in the cytoplasm. The myxoma virus is spread through insect or arthropod vectors, in this case by mosquitoes, passively, meaning it does not infect the carrier. The myxoma virus was originally a native pathogen to the South American rabbit, Silvolagus brasiliensis, where it coexisted with its host in a non-lethal relationship. It had been found that the myxoma virus could migrate to the European rabbit, where it was killing its host with extreme efficiency, likely due to the fact that the European rabbit had no natural defenses to such a disease. The effectiveness of this disease, known as myxomatosis, was one of the leading reasons it was chosen to exterminate the rabbits in Australia. However, the high fatality rate for the virus was having an unexpected effect on both the rabbits and the virus. While a large majority of the rabbits were being killed off, the small subsection of the rabbits that survived the disease were being selected for, and thus a genetic resistance to the virus was developing in future generations. Meanwhile, the high fatality rate was actually not very advantageous for the virus either. The virus was killing its host quicker than the host could spread the disease to other rabbits. This led to strong selection of less virulent strains, enabling more replication and a longer lifespan for those rabbits that were infected. After initially drastically reducing its lethality to allow it to spread, the myxoma virus continued to adapt and regain some of its virulence, to avoid being completely resisted by the rabbits. The rabbits and the virus continued to evolve alongside each other, until the rabbits had developed a relatively strong resistance to the virus, and the newest strains of myxoma were much less virulent than the original SLS strain. Within 7 years of release, the overall mortality rate was only 30%, whereas at the onset the fatality rate was over 99%. To date, the country is still plagued to a lesser extent by these rabbits, only now they are constantly adapting to the myxoma virus's attacks, developing newer resistances, while the virus is similarly adapting to stay virulent and yet allowing the host to survive to spread the disease. This continuing interaction between the rabbits and the myxoma virus is a perfect example of the idea of coevolution of host and pathogen. This has been a presentation by Jessica Doyle and Dan Zayats.